Hello students, welcome to Misostudy. In this video session, we will be discussing about the questions which are related to cell cycle and cell division. In the cell cycle and cell division chapter, we have discussed how this whole cell cycle proceed starting from G1 phase, S phase, G2 phase and M phase. How this whole cell cycle proceed. We have seen what is the difference between mitosis and meiosis. In this video session, I am discussing 10 questions, and these 10 questions I have tried to incorporate all those topics so that you can clearly interlinked with one another. In the mitosis and the meiosis, different stages we have studied, and what is the difference between the basic difference between mitosis and meiosis, what actually happens within them, we have already completed that. In this, let's start the topic, and with this, let's start the first question. The first question is based upon spindle fibers. The question says spindle fibers attached on to. Now listen to this very carefully. What we have is a spindle fiber, and we know that the function of spindle fiber is moving the chromosome, is to migrate the chromosome towards the opposite pole. Whatever the chromosome which is present in the center at the metaphase plate, at the starting of the anaphase, they will start migrating to the opposite pole. At the late anaphase, they will reach up to the opposite anaphase. They are discussing about the spindle fibers are attached to chromosome, we know that, but there is a one specific position on which these spindle fibers they are attached. Kinetochore, centromere, kinetosome and the telomere. From all of these options we have to find one. Let's understand this with the help of a diagram. This is a spindle fiber which is coming from one end, and this is a spindle fiber again. Similarly, on the opposite pole, the same type of spindle fibers, they are present. Now tell me one thing, the place from where the spindle fiber arise, this is called as centriole. So the centriole, they are present like this. They are present like this. In the spindle fiber, they are attached to one specific position. This is a chromosome. This is how the chromosomes they are arranged. This is how the chromosomes they are arranged. Chromosomes are attached at a specific position of chromosome, and that is called as kinetochore. In the chromosome, the kinetochore region is present. What are these? These are centriole. What are these? Spindle fibers of one end of one pole, and these are the spindle fibers of opposite pole. Similarly, this is also a centriole which is present at another end, and these all are enclosed in the membrane. So our motive is solved. Kinetochore is that region at which the chromosome they attach themselves to the spindle fiber. Spindle fibers they are attached on the kinetochore of the chromosome. So the correct option is option number A for first question. Let's look at the second question. In this question, I have tried to incorporate the different phases of a cell cycle. Let's match the column 1 and the column 2. Column 1, the G1, S, G2 and G0 phase is there. Let's start with the G1 phase. G1 phase is a starting phase. That means it is that phase in which the cell has already completed one of the mitosis, and now it has to enter into the second mitosis cycle. That means First of all, we have G1 phase, then S phase, then G2 phase, then M phase. This is how the whole cycle goes on. G1, S, G2 and M phase. Cycle goes on like this. Let's come at this side. We are discussing, first we have as a G1, then S phase, then G2 phase, then we have is the M phase and again. Now G1 phase is that phase in which the first of all the cell has already undergone the mitosis. Now it has to enter into the G1 phase so that it can prepare itself for the S phase, for you can say for synthesis phase, and in synthesis phase the DNA replication occur. So this is an intermediate between mitosis and the S phase. Let's come back to the question again. G1 phase is an interval between mitosis and initiation of DNA replication. That means what do you mean by this? That means S phase. S phase, right? So the correct option for in column number 1 for first one we have is a fifth option. Now let's look at the option, that means C and D, they both are correct. Let's look for the another options. S phase is that phase in which the replication of the DNA occur. This you have to remember. How you can remember this? That is, it's the S phase, that means synthesis phase. For the B, the correct option is the first option, and this one seems to be correct. So with this two only, I have already concluded that this C option is the correct option. But we will not stop here. We will discuss some more options. G2 phase. G2 phase is that phase in which basically what happens is that this is the C phase, the G2 phase, in which the replication of DNA occur, and similarly in the G2 phase, in the C phase, we have the protein synthesis occur, because the DNA is already synthesized. DNA is there, now it's time to form the protein, now it's time to form those enzymes. In this phase, the CDKs, they are formed, they actually regulate that it has to stop here or it has to shift into the another phase. So the correct option for the G2 is the protein synthesis. We are left with one. So in this is the fourth one, this is right one. Let's look at the D option. In the D option, the G0 phase is there. G0 phase is that phase that is called as a quiescent phase, in which no changes in the cell is going on. That means this is a quiescent stage. The correct option is option number 2. So correct option is option number C for question number 2. Let's do the next question. Select the correct option with respect to mitosis. We are discussing about the mitosis. That means we are discussing about the somatic cells. Let's explore options. Option A states chromatids start moving towards opposite pole in telophase. We have studied the different phases: prophase, metaphase, anaphase and the telophase. In the prophase, what happens is that the chromatin fibers formation occur, the condensation, the chromatin, they start occurring. Metaphase, the chromosome, they start arranging themselves at the metaphasic plate or the center plate. After this, the anaphase come and they try to pull these chromosomes towards the opposite pole. When the chromosome, they are moving towards the opposite pole, later on the last is called as a telophase. But the first one states that the chromosome start moving towards the opposite pole. No, in the case, the anaphase, they start moving towards the opposite pole. Anaphase. So this is the wrong option. Golgi complex and the endoplasmic reticulum are still visible at the end of the prophase? No, not at all. They already disappear. Chromosomes move to spindle fiber equator and get aligned along equatorial plate in the metaphase. Yes, we know that in the metaphase what happens, the chromosome, they arrange themselves at the metaphase, and later on these kinetochores, these chromosome kinetochore, they are attached with the spindle fibers, and these chromosomes, they are stretched, they move towards the opposite pole. That means that this is the correct option. Let's look at the D option. Chromatid separates but remain in the center of the cell in anaphase. No. In the anaphase, they start moving towards the opposite end. This is the question number 3, correct option C. Let's look at the next question, question number 4. Which one of the following event occur during the G1 phase? As I've already told you, the whole cell cycle is divided into 4 different phases: G1 phase, S phase, G2 phase and the M phase. G0 phase is a separate phase, but normally we say the 4 phases are there because we are talking about the cell cycle. G1 phase is that phase in which the cell is stable, that means no as such the DNA synthesis is occurring. Synthesis occur in the S phase, protein synthesis occur in the G2 phase. Mitosis, the separation of the chromosome, the formation of the 2 cells occur. Let's explore the options. DNA replication. DNA replication occur in the S phase. Growth and the normal functioning of the cells. Yes, this is the right option. Mutation. Is that so the mutation happens? No, not at all. It's not related to mutation. It's not at all related to fertilization. Correct option is option number B for question number 4. Let's explore the next question, question number 5. A somatic cell that has just completed the S phase of its cell cycle as compared to gametes of the same species has. Now, they are discussing about the number of the chromosome and amount of the DNA. That means A indicate that twice the number of the chromosome and twice the amount of the DNA. We have to discuss the 2 cells. One is a somatic cells and another is a gamete cells. And we have to discuss that after this S phase, what will be the number of the chromosome and what will be the amount of the DNA which is present in them. Let's understand this question with the help of one chart. Suppose this is a normal somatic cell. These are the number of chromosome and this is the amount of DNA. Number of chromosome and amount of DNA. In case of the somatic cells, the mitosis occur. Number of the chromosome, they remain same. This is also called as a equational division. Let's see, the number of the chromosome in this case is N and the number of the DNA in this case is a X. Let's do this for haploid, or they are discussing about the gamete. In the gamete, we know that the number of chromosomes, they are half in number. That means out of this whole, only the n number of the chromosomes they are present and they have an equal amount of the DNA. After this synthesis, the number of the chromosome, they remain same, but the number of the DNA, the amount of the DNA, they double, because we are discussing about these S phase. In the S phase, the amount of has just completed, and as compared to the gamete, the same species has, they will be having the same number of the chromosome, but because the S phase has occurred, the chromosome number will be doubled. In this case, the chromosome number will be doubled. As somatic cells are there, that means the number of the chromosome in this case will be 2n. Whereas in case of the DNA, that will be 2x is there. Whereas in case of the gametes, this will be n and this will be x. We are discussing about the somatic cell, the number of the chromosome in S phase and etc. The chromosome number, they remain constant. Because S phase has happened, that means the number of the DNA will be 4 times, because they will increase and synthesis of these chromosomes, synthesis of DNA occur in the S phase. So let's look for the options. A indicate the twice the number of the chromosome and twice the amount of the DNA. No, this is not the right option. Same number of the chromosome, not at all. Twice the number of the chromosome and 4 times the amount of the DNA. This is the correct option, because we are discussing about these somatic cells and we have to discuss after this S phase. So the correct option is 2n and the 4x. So the correct option is a C. Let's discuss the next question, question number 6. Now this is also one of the interesting question, so that you can clearly understand what are these stages. From these different, I can easily notice that the two things are happening within the two figures. In the first, the chromosomes, they are trying to move towards the opposite pole. That means this is a region for anaphase. That means for anaphase is occurring over here. They are moving towards the opposite pole, so this is an anaphase. In this case, the prophase condition can be seen, that means the condensation of chromatin is occurring. That means the correct option for this is an anaphase and this is a prophase. Anaphase and the prophase, right? So for that, we have to look for the option in which such type of conditions can be seen. Prophase, anaphase, no, this is wrong. The metaphase, telophase, wrong one. Telophase, this is a wrong one. In this case, it is written over here like this, but actually it is anaphase. This is a late anaphase. It is written by mistake over here. And next is the prophase. That means the correct option is a D over here. The late anaphase and the prophase is the correct option for that. Let's look at the next question. How many chromosomes will the cell have at G1? After S phase and after M phase, it has a 14 chromosome at interphase. That means they are discussing about the mitosis. In the mitosis, the number of the chromosome, they remain same. Number of the DNA increases, protein synthesis occur, but the amount of the chromosome, they remain same. If they are having the 14 at the G1, they will be having the same 14 at the S phase, same 14 at the M phase. So the correct option is option number B for the 7th question. 14, 14, 14, they remain same because mitosis is what? Equational division. Whereas meiosis is reductional division. And meiosis, reductional division. This is reductional division. Let's look at the next question. Which one of the following characteristics feature occur during mitosis in a somatic cell? Which one of the following is not a characteristic? Sorry. So we are discussing that this is not a characteristic. We know the difference between the mitosis and the meiosis. Let's look for the option. Option 1 states the disappearance of the nucleolus occur. In the this early prophase, disappearance of the nucleus and the nucleolus definitely occur. Disintegration occur. That means this actually happens in mitosis. Second option, the chromosome movement. This normally will happen. This is the right one. Next is the synapses. Synapses occur in mitosis or not? Synapses is what? Two homologous chromosomes, the pairing occur. In this case, in case of the mitosis, this synapses doesn't occur. So this is the right one because this doesn't happen. Let's look for the D option, spindle fibers. Obviously, when the chromosome movement is occurring, that means the spindle fibers are there. Spindle fibers, they arise from the centriole. So this is the D is correct, A is correct and B is correct. The non-correct option is the C, synapses. So let's look at the next question, question number 9. A cell at a telophase stage is observed by a student. That means which stage? Telophase stage. Telophase, in this the movement of chromosome has occurred. Now it's time for the formation of nuclear membrane. It's time the formation of the nucleolus, endoplasmic reticulum, etc. Observed by the student in a plant bought from the field. He tells his teacher that this cell is not like other cell at the telophase stage. Now there is a difference between the other cell and this cells. There is no formation of the cell plate, thus the cell is containing more number of the chromosome. That means cytokinesis is not occurring. They have said that this is a plant cell, and in this case the cell plate is not there. Cell plate is not there now. We can see that no cytokinesis, only karyokinesis is occurring. As compared to the other dividing cell, this would result in. Now listen to this very carefully. Cell is undergoing G1 phase, S phase, M phase. In the M phase, the interphase is happening, or cytokinesis, karyokinesis is happening, but cytokinesis is not happening. That means the chromosome number, they are increasing. But there is no separation of the chromosome into the distinct cells. So these chromosomes, they will try to accumulate like this, like this, like this only, and there will be increase in the number of the chromosome. Instead of having a one pair, they will be having a more than one pair, two pair, three pair, four pair, five pair, six pair, etc. So the correct option is a polyploidy. That means appearance of more than one or two sets of chromosome is called as a polyploid. If they have a one set of chromosome, monoploid, two set of chromosome, diploid, many set, polyploid. And we have a very good example of the wheat. In the wheat, the 6N chromosome number occur, triticum aestivum. So the correct option is a polyploidy only. For the question number 9, option A is the correct. Let's look at the question number 10. Now let's discuss about the meiosis. We have to match the stages of the meiosis which is given over there regarding pectine, metaphase, dikinesis and zygotine. These pectine and this metaphase and the zygotine, these are the different stages, and we have studied these two stages in our, you can see the mitosis also. Now how you can remember these things? First, let's see these two options, because we know that in the metaphase there is orientation of the chromosome at the metaphase plate. Let's look at the B option. Suppose I know only B and C. How I can solve this problem? Metaphase 1. In this, orientation of the chromosome occur at the equatorial plate. That means for the B, the fourth one option is correct. For the B, the fourth one is correct. That means these three might be correct. Let's look at the C option, dikinesis. Dikinesis means what is happening? The formation of the two cell is occurring. That means what is happening? We have the two cell formation. That means the two cell formation will occur. Pairing of the homologous chromosomes? No. Terminalization of the chiasmata? No. Sorry, we are discussing about the dikinesis. Right, I was confused with these dikinesis. First of all, in the pectine. Pectine is a stage in which the crossing over occur. So this is the stage of the crossing over. So the correct option for the A is the third one. That means this one is a correct option. After this, the dikinesis occur. Dikinesis is the stage of the prophase. In the prophase, the dikinesis, that means a terminalization of the chiasmata occur. That means this chiasmata structure is occurring. X-shaped chromosome, we can see that. So for the C option, the second option is correct. For the C option, the second option is the correct one. For the D, the zygotine. First of all, in the prophase, we have the leptotene stage, then we have a zygotine. In the zygotine, what happens is that these homologous chromosomes, they come together with one another, and the pairing of these homologous chromosomes occur. That means for the fourth option, the first option is the correct option. So after this matching, as I have already told you, the first option is the correct one. That means for the 10th, the option A is the correct option, right? So students, we have completed the 10 questions out of it. And these 10 questions, I have tried to incorporate the meiosis as well as mitosis. We have already solved that these are the number of the chromosome, these are the amount of the DNA. These are the frequently asked questions. Related to these, only such type of questions will be asked. So thank you so much students for watching this. We will meet you next session.